Hello dear students, today we are going to discuss some very important questions from the chapter photosynthesis. Okay, the first question is a very simple and direct question. The bell jar experiment conducted by Joseph Priestley proved that, according to this experiment, the oxygen is released during photosynthesis. That is the result of the experiment. Next question. When Rubisco incorporates carbon dioxide and ribulose 1,5-bisphosphate together, the resultant complex rapidly splits into. You know that Rubisco is a very important and the most abundant enzyme in the biosphere. Ribulose 1,5-bisphosphate is a 5-carbon compound. For example, carbon, 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 carbon. So it is a 5-carbon compound. Attached to the first carbon there is a phosphate. Attached to the 6th carbon also there is a phosphate. So this compound is called ribulose 1,5-bisphosphate or biphosphate. So when one carbon dioxide is added to it, when one molecule of carbon dioxide is added to it, a carboxylation takes place. As a result, the 5 carbon plus 1 carbon forming a 6 carbon compound. And this 6-carbon compound is found to be unstable, so that it will be broken down into 2 3-carbon compound. That means that one 3-carbon compound is like this, having 1 phosphate, and another 3-carbon compound is like this, again having 1 phosphate. And this is what is called 3-phosphoglyceraldehyde or 3-PIGA, 3-phosphoglyceric acid. This is called 3-phosphoglyceric acid. And this is also called 3-phosphoglyceric acid. It's not a glyceraldehyde, it is glyceric acid. So, when one molecule of carbon dioxide is added to ribulose 1,5-bisphosphate, the unstable hexose is produced, that is immediately broken down into 2 molecules of 3-phosphoglyceric acid, 3-PIGA. So, here the correct answer will be 2 molecules of 3-PIGA. A will be the correct answer. Next one, also a direct question. The experimental material used by Van Niel to prove that oxygen comes out from water. So as purple and green sulfur bacteria. Next question. Number of ATP and NADPH required for the fixation of each molecule of carbon dioxide during Calvin cycle. Calvin cycle means the dark reaction, also called the C3 cycle. So the total number of ATP and NADPH2 needed will be when 6 molecules of carbon dioxide is converted into 1 molecule of glucose C6H12O6. You can say that 18 molecules of ATP is needed and 12 molecules of NADPH2 is needed. Okay, so this is for 6 carbon dioxide. For 1 carbon dioxide, for each carbon dioxide molecules, so the ATP needed will be 3 ATP and the NADPH needed will be 2 NADPH. So the answer will be 3 ATP and 2 NADPH. Next question. The primary carbon dioxide fixation product in a C4 plant. You see in the C4 plant, the first cycle is C4 cycle or Hatch-Waxman cycle, where phosphoenol pyruvic acid, which is a 3-carbon compound, receives 1 molecule of carbon dioxide in presence of the enzyme peptase, producing a 4-carbon compound called oxaloacetic acid. This is the first reaction, carboxylation reaction. So the product produced is a 4-carbon compound. That is why the cycle is called C4 cycle, and the product is actually oxaloacetic acid. So here the answer is B. The 6th question. The first enzyme needed in Hatch-Waxman pathway for the fixation of carbon dioxide. The answer is already discussed. The first enzyme, that means phosphoenol pyruvic acid, is converted into oxaloacetic acid by combining with the carbon dioxide. Phosphoenol pyruvic acid is a 3-carbon compound, oxaloacetic acid is a 4-carbon compound, and the enzyme needed is called phosphoenol pyruvate carboxylase, otherwise called peptase. Answer is A. Next question is a direct question. The principle of limiting factors was proposed by Blackman. It's a Blackman law of limiting factors. Next one, photorespiration occurs. What is photorespiration? Photorespiration means C2 cycle. So here, RUBP is a 5-carbon compound, and the RUBP can undergo a carboxylation process by combining with the carbon dioxide, producing 2 molecules of 3-phosphoglyceric acid. Okay, and this is under the influence of the enzyme Rubisco. The same enzyme can combine and attach oxygen with RUBP. So when carbon dioxide is attached, the 5-carbon compound is converted to 6 carbon, then later broken down into 2 molecules of 3 carbon. When oxidation takes place, oxygen is attached to RUBP using the same enzyme Rubisco. What happens? A 3-carbon compound plus a 2-carbon compound will be produced. That means the RUBP is oxidized, broken down, isn't it? So when photorespiration, this process is called photorespiration, and this process is called a Calvin cycle. So during photorespiration, the Rubisco attaches oxygen to RUBP. Next question. The primary difference between C3 and C4 plants. In C3 plant, only C3 cycle takes place. And the C3 cycle takes place within mesophyll. But in the case of C4 plant, the first will be C4 cycle, and then it is followed by C3 cycle. And the C3 cycle occurs inside bundle sheath. Is actually the difference. So here in the C3 plant, there is the possibility of C2 cycle. C2 cycle is possible. The C2 cycle is otherwise called photorespiration. But here, since the carbon dioxide is transported from mesophyll to bundle sheath, the C2 cycle is not possible. C2 cycle never occurs. C2 cycle reduces the photosynthetic efficiency. That means the C4 plants are more efficient in photosynthesis than C3 plant, only because the carbon dioxide is escaped by photorespiration of C2 cycle. So the primary difference between C3 and C4 plant is that in C3 plant, photorespiration is possible, but in C4 plants, photorespiration is prevented. Calvin cycle, that is the C3 cycle, they take place in 2. Light reaction is an essential step in the photosynthesis, and transpiration is nothing related to photosynthesis. So the answer will be photorespiration. Next question. The site of C3 cycle in C3 plant is dash and C4 plant is dash. So as you know, the C3 cycle occurs in C3 plants and C4 plants. In C3 plants, it occurs inside mesophyll. In C4 plants, the carbon dioxide is transported from mesophyll to bundle sheath, so that the C3 cycle occurs inside bundle sheath. So in C4 plants, there is an anatomical adaptation that the enlarged bundle sheath cells are there. That modification is called Kranz anatomy or Reith anatomy. So here the correct answer will be, the C3 cycle take place in C3 plant, it is in mesophyll. And in C4 plant, the C3 cycle take place in bundle sheath. So this, so answer B. Next question. In cyclic photophosphorylation, electron emission take place from. So you know there are 2 types of pigment system. Pigment system 1, it is called PS1. And there is another pigment system called PS2. In cyclic photophosphorylation, the electron emission takes place from PS1 and returning back to PS1. This is the emission of electron. The electron is starting from PS1, emitted from PS1 and finally returning back to PS1. So the process is called cyclic. In non-cyclic photophosphorylation, the electron is emitted from PS1 and that enters into PS2 that enters into PS1 and that is released out. That means the electron emitted from PS2 is not returning back to PS2. So the process is called a non-cyclic process. So this one is called cyclic and this process is called a non-cyclic. So there will be an electron deficiency occurs here in PS2 and that is compensated from water, the electron taken from water during photolysis. So in cyclic photophosphorylation, electron emission takes place from what? PS1. Next one, absorption peak of reaction center of PS2. So there are two pigment system, PS1 and the maximum absorption, the photocenter is P700 and there is another pigment system PS2, the photocenter is called P680 where the maximum absorption takes place. So the absorption peak of reaction center of PS2 occurs at 680 nanometers. Next question. Electron emitted by the photolysis of water will be absorbed by. So here this is PS1 and here it will be PS2. So during non-cyclic photophosphorylation, electron emitted from PS2 enters into PS1, from where it is released out. This is the direction of transmission of electron. Okay, that means there occurs an electron deficiency in the PS2. It is not returning back to PS2. So this is compensated by a process called photolysis. When water is broken, we'll be getting H2 plus O, that is half O2. The oxygen is released. The H2 is again broken, producing H plus and E minus. 2H plus and 2E minus. So the electron deficiency occurred in the PS2 is compensated by the electron emitted from a process called photolysis. Okay, photolysis means the breakdown of water, the ionization of water. So electron emitted by photolysis of water, this process is absorbed by PS2. Next question. ATP and NADPH are synthesized during dash phase of the photosynthesis. You know, photosynthesis has two phases. Photosynthesis, the first phase is called light reaction, and the second phase is called dark reaction. The light reaction is a photochemical phase. And dark reaction is a synthetic phase, biosynthetic phase. Okay. So during light reaction, by using sunlight, ATP molecules and NADPH molecules are synthesized. Okay. So these are the products of light reaction. And these products are utilized for converting carbon dioxide into carbohydrate. So synthesis of carbohydrate take place in the dark reaction. So here the products of light reaction ATP and NADPH2 are used for the synthesis of carbohydrate from carbon dioxide. So according to this question, ATP and NADPH are synthesized during that phase of photosynthesis. What is that phase? Is the light reaction, otherwise called photochemical phase. Answer is B. Next question, question number 15. Oxidation of RUBP by Rubisco will lead to the formation of that you already discussed. RUBP is a 5-carbon compound. When oxidation takes place in presence of the enzyme Rubisco, the 5-carbon compound is broken down into a C3 acid and a C2 acid. That is why the cycle is called C2 cycle. So answer will be C3 and C2 acids. Next question. In a plant possessing Kranz anatomy, bundle seed cells are. So, in a normal plant, without Kranz anatomy, there is upper epidermis, the lower epidermis of the leaf. This will be mesophyll, and this is the vascular bundle, and the vascular bundle is covered by a thin layer of bundle sheath. So this is epidermis, upper epidermis. This is lower epidermis. This is mesophyll. This is vascular bundle. The covering of the vascular bundle is called bundle sheath. But in C4 plants, what happens is that this will be the vascular bundle. The vascular bundle is covered over by an enlarged cells. This kind of anatomical modification is called Kranz anatomy. Okay, the enlarged vascular bundle. So here in Kranz anatomy, the bundle sheath cells are larger. So here, the C3 cycle take place inside the bundle sheath. C3 cycle take place inside the bundle sheath. Here C3 cycle take place in the mesophyll. Okay, so when Kranz anatomy is there, in order to conduct C3 cycle inside the bundle sheath, the rubisco is present inside the bundle sheath, absent in the mesophyll. So it is larger and rich in rubisco. So answer is A. Next one. Which of the following is not a stage of Calvin cycle? So Calvin cycle or C3 cycle has three stages. The first one is called carboxylation. Second stage is called reduction. And the third stage is called regeneration. These are the three stages of C3 cycle. Carboxylation of RUBP, reduction or glycolytic reversal, and regeneration of RUBP. So carboxylation is already there, reduction is there, regeneration is there, which is not, actually decarboxylation is not a process. So the answer will be decarboxylation. Next question, photosynthetic pigments which is yellow-green in color. Yellow-green color is chlorophyll B. Next one, the primary carbon dioxide acceptor in C4 plant. In C4 cycle, we already studied that phosphoenol pyruvic acid, which is a 3-carbon compound, is the primary acceptor of carbon dioxide, get converted into a 4-carbon compound called oxaloacetic acid. That is why the cycle is called C4 cycle. So the primary carbon dioxide acceptor is what is called phosphoenol pyruvic acid, which is a 3-carbon compound, and the enzyme used here is called pepcase. So answer is A. Last one, which of the following is the major limiting factor for photosynthesis? Limiting factor means the factor which is present in the minimal quantity, lowest to its minimal value. So when we are comparing light temperature chlorophyll, carbon dioxide, the atmospheric concentration of carbon dioxide is only 0.03%. So it is in the nearest to the minimal value. So the most important limiting factor as far as photosynthesis is concerned is considered to be carbon dioxide. So we have discussed 20 major questions from the topic photosynthesis. You go through all these lessons very carefully. Okay.